Hello, everyone. Welcome this morning or this today, wherever time zone you may be, to this webinar on preparative mass spectrometry for structural biology, brought to you by the Protein Society. My name is Liz Meyering. I'm the president of the Protein Society, and it's my great pleasure to introduce our webinar today on this frontier research topic. So before turning this over to Brandon Ruotolo, the organizer for this webinar, I'm going to make just a few brief announcements. And first of all, I wanted to also mention that in terms of our webinar series, if you have suggestions for topics or if you would like to host a webinar, please do contact us. You can find information about our webinars on the Protein Society webpage for the webinars. I want to also just mention an upcoming webinar, Innovation in Protein Modeling for Drug Discovery. That will be taking place in November. And I'd like to invite you to join us for our next annual symposium in 2025 in San Francisco. We have an outstanding program, many travel awards for young scientists, and you can also find more information about the symposium on our website. And the last announcement, I want to highlight our journal of the Protein Society, Protein Science, which publishes high-quality science in a wide range of areas of protein research. So I encourage you to publish your work there as well. And so with that, I'm going to turn to our main event, the webinar. So I'd like to warmly thank Brandon Ruotolo for organizing this amazing set of speakers and for leading this webinar. Brandon's research group at the University of Michigan is at the frontier of protein mass spectrometry, developing new tools and methods for determining the structure and stability of proteins and multi-protein assemblies that are important in biology and medicine. Brandon has been recognized with many awards, including the Protein Society Young Investigator Award, and recently the Beeman Medal for from the American Society for Mass Spectrometry. So I'd also like to thank our speakers, Stephan, Rauschenbach, Charlotte Uetrecht, and Josh Coon for being here today and sharing their exciting research with us. I have just to finish off here some logistical things. So the way that the webinar will proceed is each speaker will give a 30-minute presentation followed by 5 minutes of questions. Following the talks, we'll have further questions in a discussion format among the speakers. So please take a moment now to find your Q&A tab. So with this tab, you can type your questions at any time. Only organizers and speakers will be able to see them, so they don't go to the general audience. Just a few of us see them. Brandon will read out the questions after the talks. And as there won't be time to answer all of them in real time, the speakers can also type answers to the questions in the chat. I want to thank all of you in the audience today for joining as well. Your questions and participation are also so important, and they really enrich this whole webinar experience. So please do share your thoughts, and I look forward to your participation. And with that, I will turn it over to you, Brandon.
Sorry for that brief pause. Thank you again, Liz, for that great introduction and for all those great announcements. I'd like to add my welcome to Liz's. Thank you so much for being here. I think we have an outstanding program in store for you today that really captures what I think is a very fast-moving and really exciting area, that the intersection of mass spectrometry and structural biology. And we have an absolutely fantastic lineup of speakers to tell you about this great area. So before we get into the speakers and their cutting edge science, I wanted to give a few words about my viewpoint on the history of this space. And it really goes back to World War II in my mind and the 1940s. This is where we see really the first major advancements in what I would call preparative mass spectrometry. The pictures I'm showing you on this slide are from Oak Ridge National Laboratory in the 1940s. These pictures are from gigantic mass spectrometry setups called the calutron that were used to purify uranium-235 from the more abundant uranium-238 for nuclear weapons production. And obviously, this type of technology fell out of favor for that specific application, but it sort of set the foundation for the research to come. And I would also point to this paper from Graham Cooks's lab, now about 20 years old, that revisited these basic ideas of preparative mass spectrometry, but proved that you could do this with biomolecules and use mass spectrometry in a preparative mode to purify out proteins and prove that they're active after you've collected them at the end of the mass spectrometer. And I got involved in this story as well, only briefly as a postdoc in Carol Robinson's group, who is really one of the pioneers in thinking about how this technology could be used for structural biology. The images you see on this part of the slide are from a paper we published in 2010, showing some very early evidence that you could land complicated protein complexes like GROEL and see images that were at this point pretty fuzzy, but indicated to us that there was promise in this area. And in just the last few years, we have some really groundbreaking science. These are all images from the research of our panelists today that show how far we've come in this area and why we're all so excited. The images that you see on your screen are all from data collected from ions that have been prepared by mass spectrometry, separated by this technology, but we can still image the details of these structures in remarkable ways. And that's what we're going to be hearing about today. So the schedule is up here on this slide. After I finish my remarks, we're going to have Charlotte Uetrecht from CSSB Hamburg, where she is a director and team leader of their program there, speak to us. After that, we'll have Josh Coon speak to us, who's really one of the pioneers in trying to develop soft landing technologies for structural biology application. And then we'll have Stephan Rauschenbach from Oxford, who has really, I think, pushed us into new territories with a cryo-EM image that he published in this area earlier this year. And after that, as Liz mentioned, we'll take additional questions. I want to remind all of you about the Q&A tab. Please do use that. I'll be looking at that throughout all of these presentations and looking for your questions to feed to the panelists during both the Q&A sessions after their individual talks as well as in this final session. With that, I will turn things over to Charlotte.
Thanks, Brandon. First of all, of course, for inviting me. I have to find back my slides. There we go. So while the other two speakers will talk about combination with cryo-EM or EM in general, what we do is combining native mass spectrometry with X-rays. And as Brandon pointed out, my lab is located at the CSSB in Hamburg, but we also work at the University of Lübeck since 1st of April, so pretty fresh. And I should also mention that we're furthermore supported by DESY, which is the German electron synchrotron, which we use a lot. You will see that later on. Just a little teaser that maybe some of you have seen before. It was recorded on the occasion of the Leibniz year, probably some 8 years ago. And I like to use that to showcase that viruses fly through air from one host to the other. So if we put viral particles into the gas phase of a mass spectrometer, it's not that artificial, so to say. Of course, we all know that due to the pandemic, where all of us have become semi-experts in viruses and viral spread. So just very quickly, because I will only talk about one of the things we work on in the group, We have three major research topics. One is about viral capsid assembly. Capsids are the shells that usually incorporate the genome and then shuttle it from one host to the other. And there our favorite virus is norovirus, where we, for example, look at size control of the capsids. We can get large ones out of 180 copies or small ones out of 60 copies, for example, if we do an N-terminal truncation. And we try to understand how size is controlled. And we also look at other dynamics in conjunction with ligands. Besides that, we also look at intracellular viral complexes that are relevant for modifying the host cell to ensure viruses are replicated. And before the pandemic, coronaviruses were under research. So I thought this is a great topic. And we settled in on coronavirus replication transcription complexes, where we look at processing of the precursors and then assembly of the complexes and so on. And last but not least is the branch that I will mainly talking about today, and that is MS-based single molecule approaches that we are developing. And just before I set out with this, I will give a very brief introduction about native mass spectrometry. And just want to showcase how large we can go. This is my favorite norovirus capsid protein, and in normal protein MS, you would denature your protein using organic solvents under acidified conditions. So upon electrospray ionization, you will get a lot of charges on this unfolded protein. And you can see that here in the mass to charge ratio, we have a lot of peaks ranging from 1,000 to 3,000. We have two versions of the protein, A and B, due to different methionine content. And this is clearly not informative of the protein complex that we're after. But if we use a buffer surrogate to introduce our virus protein into the mass spec, then we actually can retain the intact virus-like particle in the capsid. And you can appreciate that here, that the M/Z has increased by more than a factor of 10. We have lost the charge state resolution because we have all this micro heterogeneity, 180 copies of A and B in a stochastic fashion. But from the location, we can still say that this must be a 10 megadalton assembly in line with the 180 copy numbers. And in the lower mass range, just above 5,000, we see the dimeric building block. And I hope you can appreciate that this now also has lower charge states than what we had seen for the unfolded monomer. This is a further indication that we have a folded assembly. And just to show you that even with old mass spectrometers and the neat sample, you can get charge state resolution in the megadalton range. Here is an example from my PhD on hepatitis B virus capsids, where we could get charge state resolution for the 3 and 4 megadalton species. So what can we learn from native MS? Well, we get the complex mass, so that means we can deduce stoichiometry ideally, the amount of subunits. There are different subunits, then you can do the numbers, how many there should be. If it gets more complicated, the pure mass might not be sufficient to assign the stoichiometry. Then we can combine it with dissociation in gas phase and solution to get more insights into the subunit composition, but also to get an idea of composition, which subunits sit in the periphery and so on. And in conjunction with eye mobility, we can also say something about the gross shape and monitor conformational changes. And in native MS, usually the signal intensity quite nicely reflects the amount of protein in solution. So we can also say something about the ratios of these conformations. We can take that even further if we look at binding equilibria. If you have a ligand bound to a protein, it will add mass. So we can directly deduce from the mass spectra how many ligands are bound. And since the signal intensity is quite well related to solution concentration, you can take the signal intensity, go directly into the law of mass action, and basically get an apparent KD. Of course, it's never that easy. You have to do a few precautions. I won't go into that, but it's possible. And last but not least, you can follow the mass evolution over time and look at dynamics, for example, at complex assembly. And this is one of the showcases, or what drove me back then to go to Hamburg and go on to this project combining native MS with X-rays. So here is the example back then. So I showed you the viral capsids, and usually these assemble from the dimeric building blocks, as I mentioned. But how they get from here to the larger assemblies, the 60 mer, and then also the 180 mer, that's not depicted here, and how these intermediates look is very unclear. Back as a PhD student, I was able to propose an assembly model from mobility data that was in line with all the different assembly types or hollow spheres that we could see in the end. But this is all cut out from the available crystal structure. So how these intermediates really look, we don't know. And in case of virus assembly, this is a nucleated process, so it's very fine-tuned. Once the nucleus has formed, you're rapidly off to full assemble of the particle, which means your intermediates of interest are low abundant, but they're also very high interest for drug development. And for that you need more detailed structure. And how we hope to get to these, I will explain in the next minutes. So the idea is to combine with X-ray free electron lasers, short XFEL, especially the European XFEL, which is close to Hamburg. So normally, if you want to use X-rays for high resolution structures, then you use X-ray crystallography. You have a repetitive arrangement of your proteins in the crystal, very much like these guys here. You know, they all have the leg up at the same time. And from this repetitive arrangement, you get back reflections and increased signal. If you want to do single particles, which is, as you hopefully agree, the more natural state of a protein, you have transient interactions, you have conformational changes that are usually not well represented in your X-ray crystals. If you want to do that, you need very high intensity. And if you shoot your protein with such high intensity lasers, they will explode into a plasma, and plasma is no longer informative of the original structure. But at X-FELs, we not only have this super high intensity, but we also have very short pulses in the femtosecond regime. So the X-rays hit your protein, they're scattered off, and while they're on the way to the detector to record the diffraction pattern, this plasma explosion happens. So your patterns are still from the intact particle, and then you do it very much like in single particle cryo-EM. You record many patterns from different particles that you have to replenish because the ones you've hit, they explode. Then you reconstruct the three-dimensional scattering and eventually get your structural model. And this process is called imaging before destruction. Here you see a brief outline of how such a laser is built up. I won't go into detail, but this has potential to look at these transient or low abundant states that I'm interested in. However, if they're low abundant, if we talk about capsid assembly intermediates, then each species is far below 1%, and all the rest is your intact capsid or the building block. So you have a sorting problem. You need to acquire tons of data and then find the interesting patterns in there. With the high repetition rate of the XFELs, that becomes a huge bottleneck because the computers are not that fast. Colleagues have shown that you can successfully scatter in such experiments down to proteins the size of Rho-El. So we're pretty confident if we reduce the background that we can get to the interesting regime and use European XFEL for single particle imaging. Then to be able to solve the sorting problem and also to reduce background, we want to use native mass spectrometry and then connect that with the laser. And here you see the outline of the prototype we're building. I already mentioned native MS for selectivity and low background, and that the gas phase structural integrity is indeed maintained. Stephan and Josh have shown recently, and you will hear more about that later on. And here we will image on the fly, so there's no soft landing involved, which potentially could harm your protein complex. And I will now guide you through the setup that we have developed and then show a little bit of data. So at the front, we have the ion transfer interface. It all starts with nanoelectrospray ionization, because we know from that we have low background and also low sample consumption from the ion flux. We estimated back then with the standard source that we had used that we should be able to get 10,000 diffraction patterns in about a quarter of an hour. And you can see here that with high pressure, that's always 10 millibar to the left, we can get rid of all water or buffer clusters, and we only have the protein coming through. So if there is just buffer, then we basically have no signal, whereas we get better transmission for the viral capsids. This is hepatitis B again, and also for L'Oreal. And then we wanted to increase the ion transmission because this is one of the major bottlenecks. So we teamed up with Plasmatech, now belonging to Bruker, and they developed this transfer interface based on the Aerolens technology. We can slightly activate. We can also do some trapping for very large ions to make sure they're properly thermalized and then travel on through the downstream parts of the instrument. And if we compare the old and the new interface for several different proteins, hemoglobin, L'Oreal, and the norovirus 60mer, the T equals 1, 3.3 megadalton, then you can appreciate that the spectra look very similar, maybe a little bit more resolved with the new interface. But if you carefully look, then you may see that scale here is not the same. So if we put them to scale, then you can see that indeed we get a lot more ions out of the new interface. At least a factor of 10. I think in this example, it's even 25. So this is really important if we want to look at low abundance states. Then we want sorting, and we do that in a digital ion filter and trap. The digital ion filter is a quadrupole that is driven by digital waveforms. Why is that? These have huge mass range that you can easily adjust, so it's much easier than if you use the normal sine waves. And this has been designed by colleagues from the University of Greifswald, and it will enable us to purify low abundance species according to their MOVZ. And then following the quadrupole, we have the trap here. I just wanted to show you, this is still an example with cesium iodide, but we can indeed use the ion filter to separate different ions according to their mass. And here is the cutaway view of the trap in the cut drawing. The trap is important to time the particle release with the incoming X-rays. Because at the European X-File we have a very peculiar pulsing pattern and increase the ion density. And for the pulsing pattern at X-File, we need a trapping capacity of 100 milliseconds. Here you can see the device in reality, and here you can see spectra of L'Oreal and GDH after 100 milliseconds of trapping, and spectra look fine, so we don't have any evidence of structural damage. The next component is an ion mobility cell that has been developed with the team of Purdy Barron from University of Manchester and MS Vision in the Netherlands. And this is supposed to do conformational separation. Here you see it hooked up upfront to a QTOF system. It can be used to, for example, separate different assembly states from concana valent A. Here is the arrival time distribution, and here are the separate ones for 1, 2, and 3 mer. And we have also used it very recently to look at X-ray induced fragmentation of different peptide structures. And here you can see the overlay of ion mobility arrival times. I won't go into detail for this kind of experiment. Then follows the most challenging part, the dipole orientation region, which basically sits just around the X-ray interaction, which was theoretically proposed by colleagues from the University of Uppsala, and then the device built by Fastmatech. And the idea is to use dipole orientation to simplify pattern assembly. With that, you can then either use patterns with scarce or missing data, or you just need less patterns. And what you can see here are the results of a simulation where you have the degree of orientation. And here the unfolding, because if you ramp your electric field for dipole orientation too high, then your proteins will unfold. But there is a sweet spot where we have orientation, but not unfolding yet. And if we convert the field strengths, then this is in the range what we can experimentally do. Here you can see an example where we have the enhanced algorithm to align our patterns. This means it uses orientation, and you can see that even with a large hole in the middle, we do get a nice diffraction pattern alignment here compared to the correct solution, whereas if we have no orientation, in these cases, we don't see anything. And here you can see the original drawing of the dipole orientation finger. And I thought I had a picture of it. Maybe it's behind the sidebar. Last but not least, after the interaction region, we have a time of flight for online diagnostics developed by MS Vision together with John Hoyes, because at the XFEL, we needed something with a very small footprint, and the outcome you can see here. It's for first and foremost to control sample quality and influx, do proper selection, or know that selection is working properly. And this is the new design. Here you can see it assembled. We yet have to test it, but it's supposed to give good resolution also at low vacuum, which is important because the experimental chamber of the European XFEL has a poorer vacuum than actually our mass spec has. And here you see the prototype assembled from roughly a year ago, the ion transfer interface, the digital ion filter and trap, ion mobility, transfer hexapole. And we plan to do proof of principle experiments on the norovirus to identify symmetry. So I told you before that we have T1 and T3 particles, but in some samples, we also find holospheres that have a different assembly that is not in line with what has been proposed for viruses, which we call the unknown symmetry X. And you can see here that just from 2D diffraction patterns, the different sized particles look very different. We can also distinguish them just by size if we convert the diffraction patterns into a 1D Sachs pattern. And this triggered the idea that we actually, while we wait for beam time at the European XFEL, we could do gas phase Sachs, which then would also be possible for small protein assemblies below 100 kilodalton. So we set out doing that. First iteration was in December 22 with a modified MS Speedoc prototype as depicted here. We had a nanomate, so we have an automated easy source as you cannot enter the experimental region, the hatch, due to hard X-rays all the time. So you have to have automatic sample supply. And we wanted to use GROEL as a model protein. Here you can see the simulated diffraction patterns that we were hoping for. And long story short, this is the device as it went in, and here it's hooked up to the beamline. So the coupling was very successful. We got ions from the source all the way down to the Faraday cup. We performed Sachs measurements. We were very excited, but we could not record signal above noise because there was too much noise from the beamline. So in the next iteration in 23, we went with a shortened prototype. So we omitted the mass selection just to make sure we get as much sampling as possible. And we decided to use gold nanoparticles because due to the high density, they should scatter stronger. And we did a lot of stuff to clean up the X-ray beam. So at the detector, we only get the scattering from our samples. So we had some holes that we could shoot the laser, the X-rays through. Here's the X-rays, and here, from here come the ions. We had some metal pieces to clip part of the beam, and also a small pinhole to further clean up scattering from the ion optic, not from the ion optic, from the X-ray optics. And when we did that, we actually got very nice and essentially background-free scattering. So just look at this white space in the middle for silver bahanate in powder diffraction. This is what the SAX people like to use to characterize the beamline. So this was all nice. So we then went on to spray our gold nanoparticles. And what you can see here, this is after subtraction of the background, that we indeed got signal. And if we compare that to the simulation of the 20 nanometer particle, it doesn't look too bad. The only problem is that the scattering for 20 and 50 nanometer gold was identical. So we then figured out that likely the particles were not transmitted because we had some glitches in the voltage settings. And what we probably have measured is water clusters. Now, this sounds very disappointing, but if we can measure water clusters, which have much less contrast, then we should be able to see proteins. So this September, we went back to proteins, same setup as in 23 plus the nanomate. You see it here hooked up with yet improved alignment. So alignment went much faster. The detected distance was increased, but that doesn't matter so much. Here you see the simulations for ROEL, just in one direction, the 2D pattern. And here you see what you then would get as radial average. So that's what we expected. We also did ADH, but just for completeness. And the initial analysis, so we have to apply some masks and whatnot. You can see here in red the simulation and in black the experimental data. And with a little bit of positive syncing, you can see that the experimental data does follow this curve. It looks different for ADH, but ADH is essentially a flat line. And what we could also see that the photon signal in runs with ions was significantly higher than in runs without ions. You can also see the difference here from comparing the two proteins. So we clearly have signal above noise, and we're very convinced that we can now set out to analyze protein complexes with X-rays. So just to sum up, I hope I've convinced you that X-rays go well with gas phase ions. We want to use that to do time-resolved studies, single particle imaging of low populated or transient states. This will be possible for assemblies roughly above 100 kilodalton. And it's also possible in principle to look at unfolding. And then we're looking into gas phase SAX, which is beneficial for heterogeneous samples that are notoriously problematic in SAX. Here we have no lower size limit. And we can also do X-ray spectroscopy. I only briefly mentioned that. It's also interesting to look at radiation damage that we would expect. And with this combination of techniques, we can actually use the setup for the whole range that you may want to look at, all the way from peptides to large protein complexes. And it's the first mass spectrometer that's fully dedicated to high mass from beginning to end. I won't go into detail for these figures for time reasons. I just want to thank the consortia behind the MSPDoc prototype and my team, and the funding sources, and you for your attention. And if you're interested in the comic that describes these XFEL endeavors, you can download it here. Thanks for listening.
Thank you, Charlotte, for that fantastic presentation. We have some time for questions, and we have a couple of questions that have come in via the Q&A tab. So I wanted to read those out and get your responses. So the first is from Eric Baz. He asks, In the example with several oligomers of VP1, did you investigate the effect of different ionic strengths on viral assembly? And did you find out that pH 9 to be better in terms of the quality of the spectrum?
Very good point. I just showed one spectrum out of a very in-depth study where indeed we looked at different pH and ionic strengths, which impacts the assembly. In general, at higher ionic strengths, you get larger assemblies, so the viral particles are happier and they are able to withstand higher pH, so more alkaline pH. If we do pH 9, at low ionic strengths, then we basically only find dimer, tetramer, and hexamer, and not the proposed assembly nucleus, which is the tetramer. But that we find at pH 9. And if I would show you the spectrum at pH 7, which I actually did on the very first spectrum, then we get the 180 mer. So it's just an example that we can see these intermediates because they are more populated at pH 9.
Yeah. Another question from the Q&A tab. This one's from Sudam Boy. They ask, is it possible to determine the molecular mass of a protein which exists in a higher assembly state weighing about 2 megadaltons? It's a question about the mass range of what you're doing.
Yeah, so what I've shown you is up to 10 megadalton with old low resolution instruments, but there is nowadays Orbitrap UHMR and so on. So you can also get charge state resolution in this range. So 2 megadalton certainly is no problem. And this is exactly what Brandon is also doing, right? Looking at the different assembly states, oligomeric states, how that changes. This is the beauty of the technology, I would say.
Yeah, I had a couple of questions actually as well on your really exciting X-ray diffraction experiments. So the first is about your efforts to simulate the orientation or a dipole alignment of these ions. So I was wondering if you could give us a bit more detail on these simulations and how they were generated. When I think about gas phase ions, I mostly think about charge distributed equally over the surface of these ions. So it's hard to think about what a dipole might look like there. So could you talk a bit about that?
Yeah, so what the colleagues in Uppsala have done, basically they looked at the charge states that you would find from electrospray ionization for the different proteins they looked at, and then placed the charges in the most likely spot. This is, of course, a little bit artificial, and I did not mention that, but this is why the dipole orientation is the most challenging part. Because if the charges sit in a different position all the time, then orientation will not be reproducible. Then we would need to work with charge reduction. And we all know that protons are able to hop. What I take the optimism from is simulations that Lars Kronemann has done, where he actually sees that the residual water during desolvation actually clusters at certain areas, which makes sense. A water molecule would not be happy alone. It's energetically not favorable. And of course, the ionic species are dragged alongside. So there is a certain chance that that charges will be represented in one space of the protein more than in others. And that also makes sense if you think about how many charges we have. I also learned that they're equally distributed, but if you do a back of the envelope calculation for a protein of, let's say, 50 kilodalton, the charges don't really see each other if they're equally spaced, so they can as well sit closer to each other. Was a long question, long answer, but I'm not even sure I answered your question. But, that's my take.
I think that's great. It's nice to hear you thinking about these things because they're, I think, really important. So I think we have to move on, but Charlotte, you should know that there's at least one additional question in the question tab. So we'll get back to that after that, or you can take a look right now and answer. So we'll move on to Josh Coon's presentation. So Josh is currently a professor of chemistry at University of Wisconsin-Madison, and is also associated with the Morgridge Institute there in Madison. And Josh, your presentation. Looking forward to it.
Okay, thank you. Should be coming up. All right, can you see it all right?
Yeah, we can see it just fine.
Thank you. All right, thank you, Brandon. Thank you for organizing this great meeting. And it's my pleasure to talk about our efforts to do soft landing and cryogenic landing of protein-protein complexes and to try to find a way to couple mass spectrometry with electron microscopy. So for those, just 1 second, thank you, I had to move that out of the way. If you're not familiar with electron microscopy, it's becoming or has become the leading technology to do structural biology. And just sort of a simplified explanation of how it works in case you're coming from the mass spectrometry side, is that you have particles. In this case, I'm showing you elephants for illustrative purposes, but you have your protein complexes embedded in a thin film of amorphous ice or vitreous ice. And electrons pass through that, and then you can collect two-dimensional images of those particles that are embedded. From the two-dimensional particles might look like this, so you might get front views of the elephant or side views. Here's a top view. You would then do data processing to group those into what we call class averages. So here's a group of front views, and here's a group of side views, and so on. From those averages, we call class averages from 2 dimensions, you can do some mathematics to transform this into a 3-dimensional structure. So that's the rough sort of high-level way that we can get structure from cryo-electron microscopy. Now, one of the key components of cryo-EM and what made it become so useful for structural biology is the ability to vitrify your sample in water. And here's a nice review on that by Yifan Cheng and co-authors. But this is one of the early devices for freezing and preparing your sample. Just to give you a little more details about how this process works, you have your complex embedded in in an aqueous solution. And if this is our EM grid, which is obviously thicker than it would be, but so you can see it, you pipette the particles in aqueous solution on the grid. And then there are basically filter paper that comes in from either side that presses up against the grid and wicks away the bulk of the water and the sample, leaving just a thin film of water and sample in these holes in the grid, which you can't really see here, but you'll see in a moment. That grid is then plunged into liquid ethane, which is cooled with liquid nitrogen. And the idea here is that we can take away the heat so quickly from the grid and the water that you freeze, but you don't allow crystalline ice to form, but rather glass-like ice or vitreous ice. That's important because if you had crystals, you would diffract the electron beam, and that would be difficult or impossible to get images from. So you have to have this perfect kind of ice, and you have to have your particles suspended in that ice. Here's a cross-section of what that might look like. So here's a grid. This is the metal support of the grid, and then you have this carbon a mesh over the top of it. There's holes in that, and in these holes you have the ice forming, and that ice suspends the particles so that you can then pass the electrons through it. Now, the ice shouldn't be much thicker at all than the particles because you have to get the electrons through, and so you really want ideally very thin ice. So in the way I've drawn this here, this is quite thick and probably would not be ideal. So here's getting to that point of desiring thin ice. And if you have very thick ice, then it's difficult to be able to see the particles and distinguish it from the ice, and you have lousy contrast. So here's a picture over here, and you can see there's some particles in there, and the ice is around them. So that's a really key component of having successful cryo-electron microscopy and getting high resolution structures. It's this process of how you actually make the ice. Now, this is a field of study in the cryo-EM space, and there's lots of tricks and tips and methods to get perfect ice and get your particles in there. This is a screenshot from a manuscript that shows all the different ways you can have bad ice, and there's probably more than this, but there's all kind of things that can go bad, and some of them are depicted here. You see all these different shapes and particles being aggregated or strung along, or particles going to the air-water interface, which is very problematic and can cause problems as well. So just to summarize the challenges of ice making and this technique of plunge freezing that's really become an essential part of how we do cryo-EM, is that you often can have preferential particle orientation. I'm showing that here with all these particles sitting in one orientation. That's a problem because when we want to do three-dimensional solve the structure, we need to have many different views so that we can get all of the aspects and get the highest quality reconstruction. So particle orientation is a problem. Varied ice thickness is a problem. And then another major problem is this air-water interface issues that can occur, and that can cause distortion of the particle where it has frozen. So what we have been working on is to try to find ways that might eliminate some of these challenges, but also combine the benefits of mass spectrometry. And we call this matrix landing here, where we might imagine landing particles on a thin film of a grid and then coating them with ice, as shown here. We would have random orientations of the particles, and we would have them all in the same focal plane, and we would be able to control the amount of ice. So that's the ultimate goal of what we've been working on now. So that sort of gets us to this nice slide from we've adapted from a Thermo Fisher webinar. And this just sort of shows that there's a lot of biophysical technologies that people use to look at structural biology. And you have the cryo-EM method that I've just gone over here on the one end. And on the other is native MS. And we've already heard from Charlotte some of the ways that native MS can help structural biology apart from structure. But you can get all this type of information. And so we had thought several years ago, wouldn't this be a really cool idea to couple these methods? We were inspired by the early work of Carol Robinson's lab that Brandon pointed out, and some of the work that Stephan had been doing at that same time as well. Could we come up with a way to do this? So I'm going to walk you through sort of our approach to this problem and where we are. So we started with a simple experiment, which was just to take an ultra-high mass range orbit trap and just put a TEM grid on the backside where the HCD cell would be, and make it so that particles could either be mass analyzed in the orbit trap, as shown here, or landed on that grid. And then this wouldn't be directly imaged by cryo-EM, but we could just start with negative stain TEM, a lower resolution technique, but much easier to do, and just to kind of see where we are. So here's some of the early images that we acquired when we landed the GROW-EL. The particles here's what they should look like. Here's their mass spectrum that we acquired, and you can see some splats on here, and this is the experiment basically over here. We had the good fortune of hiring here in the biochemistry department Professor Tim Grant, who's a cryo-EM expert, single particle expert, and we're pretty proud and said, "Hey, Tim, what do you think of these?" And he didn't agree. He said they were not good. And so we kind of started to think about what might be happening on that surface and to these particles. And we got into a lot of the literature from the 80s and 90s and even early 2000s about what happens to proteins when they're lyophilized, and how you can, when you freeze dry things, how you can stabilize them. And here's this manuscript back in some negative stain work talking about you can reduce dehydration-induced flattening or problems with molecules by adding glycerol or glucose. That's actually an idea that some early soft landing work in mass spectrometry had used, and there's this early paper by Gary Shustak that used some glycerol, and Cook's, the paper that Brandon referenced at the beginning of the introduction, actually used some glycerol. Now, these folks landed in the glycerol liquid and then pulled that out and then tried to extract the protein from that. Our situation's a little different. We want to image right straight from that. So we experimented with very thin films of glycerol. You wouldn't even know it was on there, but putting on very small amounts of glycerol and landing on that. And what we found is that if we did that and then negative stained, we could image right straight from the landing grid. Now you can see the GRO-EL structure is evident from these landed particles in negative stain. So this is an end view. You see the hole in the middle, and here's side views. You can see the 4 rings stacked together on that GRO-EL. So we did that for, in this case, I'm showing you 3 different particles that were put through the mass spectrometer, landed either, in this case, with the matrix, or in the bottom, I'm showing you what it looks like if they're pipetted in the conventional prep. And you can see these particles look very similar, a little bit lower density here, same here, and same for these beta-galactosidase particles that we were imaging. We used, we solved 3D structures of many of those. Here's an example of the the GRO-EL particles from one of those very early landings. And we were able to get a 3-dimensional structure from the landed particles by negative stain TEM. It's about the limit of resolution you expect to get from negative stain TEM, and we were able to achieve that. Here's the structure, crystal structure. So really excited about this data because it shows us that at least to the resolution of these measurements, that the structure can be largely maintained and not lost in the transit through the mass spectrometer and in the deposition process. So that there's a movie. I should have flipped the button a little earlier, but we just talked about that. Okay, so here's a comparison of the landed GRO-EL structure to a pipetted solved one. You can see they're very similar with just a few minor differences in density here. We did some work after that to look at what other matrices might work. So the glycerol did have some challenges that it took some pretty dialed in capability to make that work routinely. We found polypropylene glycol was a bit easier to use and would allow us to have more reproducibility. So thin films of polypropylene glycol on the grid. In this case, I'm showing a very highly dense landing, probably too many particles here, of GRO-EL, and you can see some nice class averages here. Here's a 3D structure solved from this we call matrix landing method of beta-galactosidase at about the resolution you'd expect from negative stain. Here's the 20S proteasome from the same landed particles here. So just to summarize that work, the landing matrix, we believe, likely protects these desolvated protein cations from vacuum exposure. It could be that it helps soften the landing, but I think it's more about providing some sort of stabilization force on that grid, because we land for up 5 to 10 minutes. And during that time, the earliest arrived particles are exposed to vacuum for some time. So I sort of think of it, you can think of it like this: if you have something on the particle or that matrix on there, you can kind of keep the particle happy. Whereas if it's not, you kind of have loss of quality of the structure and it degrades. So this is the experiment again. And one of the reasons that I didn't highlight initially when I was telling you about the potential benefits of coupling mass spec to EM. We talked about all the issues with ice making, but also if you have a mass spectrometer in your preparation method, you can use it to purify things. So here's a mixture of the two particles I've shown you. We can land, and you can use the quadrupole to filter one from the other, and preferentially land from mixtures. And so you can see this, so we skip that one, but basically here's the idea. You've got the mixture, you can land that as a mixture, and you can see the beta galactosidase, you can see the GRO-EL, and you can choose one or the other and get highly purified particles on grid. So that's an exciting additional use of this potential coupling. Here's some structures from those landed particles. Okay. Now, what we ultimately want to do is to be able to, I guess we can go back here. We don't want, if we want to do cryo-EM to get the highest resolution, it's a challenge for us to have that matrix on the grid surface, because that causes contrast issues with the electrons. So we would like to get rid of the matrix, the glycerol and the propylene glycol. And but we've sort of concluded we need something to protect these particles. So ideally, that something would be water. And what we'd ultimately like to do is to be able to add water or ice to the particles once they're deposited in a controlled way, kind of like these trees get a thin shellacking of ice in a spring storm. So Mike Westphal in the lab had worked out a way to add water. So basically the idea is you make a molecular beam of water, and you put in water vapor, and you have a microchannel plate, and you have single water molecules coming out of that and traveling in the direction of the holes in the plate. But basically you can orient that towards your grid. If you hold the grid at very cold temperatures or minus 180 or lower, you get thin films of amorphous ice that can grow. So this has been worked out in the physics community to study amorphous ice in the lab. And we adapted that to be able to add ice in a controlled way to a landed particle. Here's an image of some ice over top of one of these holes where we had a thin film of graphene or carbon to land on. And you can see from this diffraction pattern, we have amorphous ice. So now we want to take that same experiment that I showed you, and we're going to be landing particles, but now we're going to be landing on a cooled grid where we also will add this molecular beam of water to coat the particles with ice. So it's a lot more difficult experiment to do than the one that the room temperature experiment, because we've got to cool the grid, we've got to add ice, and we've got to do that either conjunction or in sequence. So here's one of our earlier apparatus to land. And you can now see the UHMR instrument has this landing device in the back, and we have liquid nitrogen flows through the grid landing holder to keep it cold, and then we have to have a gate valve here to close it off from vacuum because we need to get the sample out. I can show you. So this is a more current image, and you can see this is a cup here that would be filled with liquid nitrogen. So when we get finished landing, we can drop the grid out and keep it under liquid nitrogen until we can put it in the microscope. Here's a blow up of the setup. So here we've put the HCD cell back in the instrument. Ions come through here, and then the grid would go in this holder, and you can see this little bit right down here is to scale. That is the actual landing grid. It's held in this device, which slides in there, and then that's all compressed up against this block, which we can regulate the temperature of. And then the water beam doser comes in, and you pull this back and is moved in front of the grid to add ice once the landing has been completed. All right, so I'll just show you some images of cryogenically landed versus room temperature landed. So here we have beta galactosidase particles that we landed at room temperature on that system, and then just directly cryo-EM imaged. And on the right are beta galactosidase particles that have been landed and ice added to them. And you can see that it doesn't look so good here. But when we do class averaging, you can see these particular particles have, you can actually see some detail in these these class averages, whereas over here, you might be able to tell there's 4 subunits, but you don't have that kind of detail present in them. So we definitely appear to be having a much higher quality particle when we land cold and are able to coat with ice. Now, when we did 3-dimensional reconstructions of those, we can see a much better particle reconstruction when we have the cold landing than when we have the room temperature landing, but it's still not at higher resolution as as could be achieved by cryo-EM. We'll talk about that in a moment. We do see when we land cold, compared to room temperature landing or conventional prep, which I don't have here, and I apologize for that. This is a distribution of the orientations of the beta-galactosidase. You can see a much better sampling of all these different angled orientations. When you land at room temperature, you get a highly preferred orientation, which indicates to us that things can move around on the grid. And when you prepare conventionally, you get more preferred orientation. So this is one of the benefits we expected from a cryogenic landing, and we're seeing that in this particular case. These are particles of the 20S proteasome landed room temperature or cold. There's looks like clearly less ice on this particular specimen. But again, we're getting higher quality particles in the cold landing, and you can start to see some of the structure in these 2D class averages, whereas here you just kind of get the shape. And again, this is a pretty lousy 3D reconstruction, but it's definitely much better than room temperature, and it's got the overall shape here. Now, one of the things that sort of we've noticed in an enhanced preservation when we cryoland, we think that the ice probably helps reduce radiation damage when we're doing EM imaging, probably reduces particle surface interactions, and maybe there's reduction of some dehydration while on the surface. So if a particle comes in with some water left as it transited, at least maybe it can retain that water and stay happy. One interesting artifact that we've noticed is that if you look carefully at these 2D class averages of the proteasome, you'll see this sort of halo around here. You see it here. And this indicates that the ice that we're putting on may not really go around like that, coat it like an M&M like we maybe have thought. And we took some tilt videos. So I just want to draw your attention right here. So here's a particle. You can see it come in and out of focus. You can see the little hole in it right there of a proteasome. And then you can see this big ball on top of it. It appears like, and there's several examples I've highlighted here if you focus on it, but what we think is happening is that these particles, when we're adding ice, it doesn't necessarily coat them uniformly, but maybe it's growing sort of structures on top of the particle, which was really not anticipated as one of our current areas of focus. How is that ice being integrated into the particle? And it may not be really going on as we might have expected. So that's something we're currently looking at. And then I'll just wrap up and say, how do we further increase structural resolution? So I think you're going to see from Stephan's talk next, he's managed to do a bit better in resolution than we have. And so with a similar approach, things that we're focusing on are trying to reduce dehydration during the mass spec experiments. I think that as they transit, they're losing water. If we can prevent them from losing water, that will help preserve their structures. I think there's a lot of interest in what our grid surface temperature is and how we're forming ice. We're looking into that quite a bit, and we're doing lots of experiments there. Stephan and I have had lots of discussions about deposition energies, and I think while we don't think that's the source of where we're getting some deformations, but it's certainly possible that we need to more gently land them. And so we're looking at that as well. And I think if we could find chemical matrices that are more compatible with cryo-EM, that would be a way to solve this problem. And then ultimately, you could even freeze them while in the gas phase before they have had a chance to lose the water. I think that's a potential solution. So lots of space here, I think, to ultimately achieve this. So the bulk of this project was initially led by Mike Westphal, shown here. Mike retired last December. So we've got a new lead not shown here, Scott Quarmby, who's been leading the project. All these images came from either Kenny or Austin. Austin's a student who's about to graduate, and Kenny was a postdoc who's now on the faculty at Brigham Young and is leading his own team. I finally just want to thank National Institutes of Health, in particular this R35 grant that has allowed us the flexibility over the last 5 years to really pursue this, what I would say is a high risk, but hopefully high reward area of research. So thank you all for your attention, and it's a pleasure to be here.
Thank you, Josh, for that fantastic talk. I have a question for you from the Q&A tab. Nicole Wagner asks, does the quadrupole on your instrument have the resolution to isolate a single charge state? I wonder if you could characterize potential subtle differences in protein structure with respect to charge using EM.
Yeah, it's a great idea. We have had a difficult time with the limited resolution of the quadrupole on that system to be able to do that experiment effectively. So no, we haven't done that. I think that's a cool idea. Would be something that I think is certainly worth doing. I'm not sure if the quality of structures we're getting right now would be able to discern differences in those, but I think that once we work that out, I think that's a goal that we have, to boost the resolution of the quads and really try to tease those things apart.
So I had a question for you, Josh, as well. I mean, you list a lot of, I think, great ideas towards the end of your presentation in terms of where to go with this technique in the future. I was wondering if you could give us a bit more insight in terms of what you think is the most important immediate goal for this field in terms of what we need to work on achieving, and how you think that can eventually be realized.
I think for our team, it is to get the higher quality structures out of the method, and I think until we can routinely do that on whatever particles we put in, then we kind of have a limited, we're still not, I mean, ultimately we'd like to see the merging of this is how you could best prepare your sample for EM. But until we can master how we preserve structure and get high quality three-dimensional reconstructions out of it routinely, I think we're not quite there. Short of that, I think addressing questions about gas phase structure is something that we're well positioned to do, but for us, I think working out the details of how to do the best preservation and boost that resolution is our #1 priority.
One more quick question from the Q&A tab from George Rodriguez. Were you reporting negative staining at room temperature TEM? I guess the question is, when you're doing the negative stain procedure, were you doing that at room temperature for your landing experiments, or were you doing that at some altered temperature?
Nope. I think all of the negative stain results I've shown today have been from landings done at room temperature. And then we just pull them out, stain them, and image them. Yeah.
Great. Okay, with that, I think we'll move on. But I think there's maybe one or two additional questions in the tab for you, Josh, once we get there. Okay, so next up, we have Stephan Rauschenbach, who's an associate professor at the University of Oxford. Stephan, the floor is yours.
I'm here. Can you hear me nicely?
Yes.
Very good, very good. Let me briefly share my screen and slides. And first of all, thanks for organizing this. Apologies that I messed up the schedule. I was sitting around at airports for way too long, much longer than I anticipated. And yeah, so it will get worse, because if you sit around at airports for a very long time, you have ideas what you could explain. And so the number of slides has been rising while I was sitting there. So I hope you can see my mouse. I hope you can see me nicely. So yeah, thanks for waiting up for me. This is the title slide. wonderful to see these two exciting experiments before and how things are progressing. And I think it gives a good feel of how complicated this is. And I think this is the beauty of this experimental direction, that you have to mix things from physics, from chemistry, from biology, all at the same time and get it all right. So so this is in my opinion, a very special, it's a very special direction, a very exciting field to be in, doing that. Yeah. So I want to start with showing you a picture of my college. So this is Wadham College. We started this meeting series 2 years ago. There, it's not that long ago, and things developing really quickly. And again, I can point out here perhaps that this is also the college of Robert Hooke, and Robert Hooke is well known for the laws of springs. So if you want to calculate forces of springs, and they will annoy you in school with it, but he's also was quite instrumental in developing practical microscopy equipment here. Many of you may have seen these famous prints or the drawing of this microscope that is in his book, Mikrographia, still shows a microscope, how we use light microscopes today. So he might not have invented this, but he made it really practical, and he wrote this book, Mikrographia. And I can only recommend everyone to just read it, because he in a tour de force, it goes through everything you want to put in a microscope, from pollen, insects, plants. He sees the first cells and calls them such, to crystals and snow and ice, and in the end he turns it around and looks at the moon and looks at small structures there. So here in Oxford we are as lucky to actually have the original version, and a while ago I took my group for motivational reasons to see this, and it's really inspiring. When you think of this 400 years ago, people for the first time saw things at that detail, and I think that's the beauty of microscopy. And you can read the book. It's actually in plain English, and I can recommend at least reading here and there. It meanders a little bit, so it's a lot of text, but you find interesting things. So for instance, this here about his issues in getting ants under control for imaging them. So he would usually use some glue or wax to immobilize them, but as you can read here, it would twist and wind its body, so I could not get a good view. And if you kill it, then the shape is spoiled. And so he concludes that this is the nature of minute bodies, that as soon as almost their life is destroyed, their parts immediately shrivel and lose their beauty. And you saw with Josh, this is exactly what we are struggling with, how to get this under control. So Robert Hooke had alcohol to the rescue here. So he could actually probably get the ant drunk and then get a good picture to draw them. And we will not use alcohol, we use other things. And so what I want to do today, originally this was intended as the first talk, I wanted to talk a little bit about the different methods we are using. So here I start with scanning probe, but also electron microscopy, and show you what is actually the problematic part about it. So what are the issues we want to solve and want to fix, and where they come from. And then show you how we fix them, so a bit more technical side maybe, and also show you a few examples. So I have to look for the watch a little bit. So for scanning probe microscopy, this is where we started this one. This is a technique which inherently delivers atomic resolution, because at this scanning tip here, there is one last atom, and this one last atom basically carries all the tunneling current. And as it does carry all the tunneling current, it immediately resolves surfaces at atomic resolution. So here the first image that became well known, that led to the Nobel Prize, together with Ernst Ruska for electron microscopy, by the way, is at atomic resolution. It shows you that this is not always so simple, that it took almost 30 years to get molecules at atomic resolution, because here people had to do a lot more tricks in the sense that they had here to attach a carbon monoxide molecule and use Pauli repulsion imaging in non-contact AFM to get these structures. So why is this the case? Tunneling microscopy images electronic structure, not necessarily bond structure. Electronic structure means there is a tip, it's a metal, it has electronic states everywhere, filled up to Fermi level. But on the surface, there's a molecule, and a molecule has some molecular states. And these molecular states have expanded in space, and molecular states will also mix. They will mix with the states of the surface. So when these states here participate in the tunneling, you never see this one single state that belongs to the bond. You see a mix of states. So basically, you're measuring the electrons near or around this Fermi edge, and these are smeared out across the whole molecule. And so normally when you look at a molecule, you see a blob. I will show you a lot of blobs in a moment. So only when you go very close in Pauli repulsion here, push this p orbital that stands out from this oxygen atom here into the carbon atom at very high energy, only then you will see atomic structure here. And you see this precision that you have to handle these samples at this atomic precision is also what complicates this application. So you have to use this method in ultra-high vacuum on single crystalline samples. So these are cleaned here. So this is a platinum sample here, heated to almost 1,000 degrees in order to recrystallize it and make these perfectly flat samples. And only then you bring on the molecules, you sublimate them. And this cleans the molecule up. Sublimation temperatures are chemically specific. So at one sublimation temperature, you get one molecule, and this will lie on your surface and you can image it. And immediately it's clear that more complex molecules, especially biological ones, they don't have vapor pressure. They will not go into the gas phase. And so if you heat up egg, you don't get gas phase egg or gas phase protein. But you burn something, you just get a lump of carbon. So they are not stable at these high temperatures. And this was for many, many years limiting this application. So let's look at the next method. So you heard we are all interested here in electron microscopy, especially in electron cryomicroscopy, because this shows us it can be used also to visualize biological molecules. In contrast to STM, electron microscopy is quite an old method, 100 years old almost here. You see Ernst Ruska built the first version in 1931. And if you look at how the electron beam moves through, you will see that it behaves very similar to an optical microscope. And so the limits, the upper resolution limit also applies to electron microscopy. The fortunate thing is that for electrons, the energy informs the wavelength. And so for the typical energies of an electron microscope, we are in the wavelength of picometers. But now if you look through my list here, you will see that I wrote atomic resolution for electron microscopy in the 1990s, because the lenses, these magnetic lenses, are really bad. People compare them, if you build a microscope with the bottom of a champagne bottle as a lens. That's about the quality that an electron lens has. And so only these aberration-corrected lenses led to atomic resolution here. So what are the issues then with electron microscopes? So it seems everything seems to be fine. And indeed it is. If you take a silicon sample, you get here these atom dimers separated by 78 picometers, perfectly resolved. So this is silicon lattice at 110 direction. And there is quite important application also here on the right side. I find it always fascinating to look at these. So this is a cross-section through CMOS memory, so what we all use in our computers at a relatively recent 7 nanometers node. And you see down here, this is the memory, and all this other stuff is just the contact layers. So people use these microscopes. They are extremely useful also in biology already. So where are the issues? So the issues start here. The electrons need to go through. It's a transmission electron microscope. So the samples need to be thin. And the traditional ways to achieve this is honing, so polishing. So this is like when you sharpen a knife or a razor blade, you start by polishing mechanically, with polishing to grit 10,000. So if you sharpen your knife sometimes, you know, this is what some people have in their households. But then it's by no means thin enough. Then you're in the micrometer range. You need to go below 1 micrometer in order to get through with your electrons. And so then argon ion beams are used for ion milling. And this is an extremely tedious process, and I've seen it go wrong many times, and people losing their good behavior over it and throwing samples in the corner when you mill a little bit too much and your sample is gone. It can be quite annoying. And so this is how you prepare these inorganic samples, like the silicon sample I showed you before. This was made in exactly that way. So what else is going on? So here, this is a very busy slide, but it basically wants to show you 2 important things about electron microscopy. And these are about the interaction of the electrons here with matter. So the electron has a charge, and it has a mass of 10 to the minus 4 atomic mass units, so 1,000th of an atomic mass unit. So an atomic mass unit has some nucleons in the nucleus, and it has a charge of Z. And so now we can look at 2 quite important properties. So first, how well do we see an atom? And for this, we can just take a look at the Rutherford cross-section. And you can see here the Z is in there. So the charge of our 2 particles that are interacting, and it tells us we see especially heavy things. This is not nice for organic molecules. There's a lot of hydrogen in there, especially hydrogen we don't see well, and actually also carbon we don't see very well. And then there is something other which compounds on this problem. There's a so-called ejection cross-section. So the ejection cross-section happens when my electron collides here. It loses some energy to the atom. You can use this to get chemical information, but it can also be enough to kick out the nucleus. And then you break a bond. And this, of course, is a big issue for molecules. And you can very simply calculate this. So you can calculate these kinematic factors here. This is basically school physics, just central collision is enough. And you will find actually these kinematic factors are small, so you lose very little energy. But then think about, you come with 100 kilo electron volts, and you need to transfer something like a few times bond energy to kick out the nucleus, which is 50 electron volt. So this is exactly in the range. And then here, Eleanor Besley in Nottingham, she calculated this once properly. And you see the bad news is, here the masses of particle and target play a role. And for light targets, you get extremely high ejection cross-sections. And so now what does this mean in combination? So if we have organic molecules which have a lot of light atoms, we don't see them very well, but we destroy them very fast. And so this leads to cryo-EM with its special ways of preparing things. So for molecules, Josh already discussed this, so I can skip through this quite quickly. You can do a liquid phase preparation, creating either very thin liquid films in plunge freezing, which embed the protein, or embed the protein with something heavier so that you can see it nicely. That would be staining. And then apply single particle averaging methods. And with all the problems that Josh already introduced, it is, of course, quite important to see that it also works really often and really well, and you get angstrom resolution if you find enough particles and average them. And to find enough particles and average them, you need nice grids. So you see here, the issues lie completely different than for scanning probe microscopy. So in scanning probe microscopy, certain molecules will not work. Here the issue is rather that I need this averaging, and I need a lot of really nice and well-prepared particles. I can even go outside vacuum. A third method that we are using is holography. It's very close to electron microscopy. It also uses an electron source at very low energy, only 50 electron volts, so there's 100. There's no risk of kicking atoms out of our object here. It is in very close proximity. There's no lenses or nothing. And then at a few centimeters distance here, you observe these holograms. And so these look like these rings here. So I always compare it to throwing a stone in a pond, and then after a while you see the waves traveling outwards. That's more or less what you have. So these rings here are a mix of a reference wave, so the unscattered wave, with the wave scattered by the object. And then in the calculation, you turn around time, you propagate all this backwards, and you get back the shape of your object. So this was proposed by Danish Garbo in the 1940s. Here, this is the sketch from the original papers. This is how it is realized nowadays here, the field electron ion microscope from these very sharp tips from the lab from the Stuttgart colleagues. And this is how these holograms look like and the reconstructions. So the issues here are again at a different region. So the issues here is you need these extremely perfect, I go back one slide, you need these extremely perfect substrates. You see an electron wave needs to pass through without being scattered. Part of it is scattered. So that's quantum mechanics. It actually happens at the same time, scattered and not scattered. And then this mixes on the surface. So you need, for this non-scattered part, to maintain a coherent wavefront, you need a perfect graphene layer. Yeah, and Jean-Nicolas Rauschenbach, back then in Hans-Werner Fink's lab, who developed this method, also developed a graphene method to produce this. So you basically fish out graphene that was created on a copper layer here, and you clean it, and you deposit it then on a platinum layer, and then you heat it up and bring it into vacuum. And this heated up platinum removes the last remnants on the freestanding graphene. So that here you can see this quite nicely. So this is the graphene, but you don't see the graphene, you see the PMMA, so the photoresist. And if you activate the platinum, you see it cleans up all the photoresist, also here reflected in these pictures. So if you then take these, and so this is what we made back then in the lab in Stuttgart, and look in the electron microscope, you get these perfect layers of atomically resolved graphene. And now you can imagine if you place a molecule on there, you will be able to apply this holography method to this molecule. And so now we find the next issue, so how to place this molecule. So you can try to place it from solution. So this works if it's something really big, like the colleagues in Zurich did this with this tobacco mosaic virus or with nanoparticles. But if they are smaller, like a single protein here, this will not work. Then solution contaminates everything, you lose your coherent wave, you don't see your molecules. Another alternative is just have a free-hanging DNA, but this causes field distortion because there's voltages applied, the electron is charged, and so this acts more like a lens. So it is attractive, the electrons bend around. It gives you a diffraction image. You can reconstruct it, but you cannot trust the dimensions here anymore. So when we put this together a little bit, then we see there's a lot of different methods that give you very different problems. So scanning probe microscopy is limited with thermal sublimation, you need these clean surfaces. Similar for holography, you need these very clean surfaces, and bring them into vacuum, maybe even more than in SPM, but you want to have bigger molecules native. And in cryo-EM, you can actually do liquid phase preparations, but you have issues with how the particles are distributed, and all of these, and how homogeneous and how clean this is. And all of this points you in the direction that you need some method that gives you a lot of control in how to put your sample where you want it, how much you want it, which type you want, no contamination in exactly the right environment. So basically what you want is control. And to this end, we want to use preparative mass spectrometry. And so when you start at the very beginning, you start in World War II, where people used mass spectrometers to enrich uranium. So they did this a lot, kilogram scale, but there is not much control to it except chemical selectivity. And then the method was forgotten for a long time, and and later came back in the 70s under the title of soft landing. So instead of just controlling chemical selectivity, you also will control the molecule or ion surface interaction. And here I picked ion surface interaction. Ion surface interactions are plenty. So you can characterize them by energy from below 1 electron volt. This is almost what happens in a gas collision, to mega electron volt. So here on this end, this is what nuclear physics does. Rutherford backscattering. Then here's ion implantation and sputtering. We all know these processes. And then here in the range of 1 to 100 eV, you get soft landing and reactive landing. These are the processes that are interesting here for what we have in mind. I think this little simulation here of different particles, so this is for a sputtering simulation in a SIMS, with a gallium atom or a C60 molecule or a huge argon cluster, nicely underpins what happens in the higher energy range. So it can be really destructive, but these are 25 kilo electron volts. And so for these lower energies, you're in the range where quantum mechanics plays a role, and where you have to really look and think what happens, but in terms of chemistry, not in terms of nuclear physics and high energy collisions. And so after a long time, after the Second World War, uranium enrichment, I think the first one I find always is Graham Cook's in a paper actually from the year I was born in, 1977. And in this paper, he builds this simple apparatus with an ion source and then landing on a metal target to look at chemistry. But he points already out he wants to have energies in the range from 10 eV to kilo electron volt, and even at these low energies he can observe chemical reactions. So there's a lot of interesting processes happening. And with the advent of electrospray ionization, maybe I don't have to explain this again, it gets a whole lot more interesting because you get a lot more interesting molecules here. So with electrospray ionization, I heard we saw this paper already here before. You can do this now with complex and biological molecules, and the inspiring experiment here was in 2003, the deposition of proteins, retention of their chemical structure, and also detection of some function afterwards. So these processes can really be gentle. And I would also like to highlight then the more sophisticated instruments here. This is a machine from Julia Laskin's lab, where they did landing with peptide ions, and they really used the kinetic energy to look at the processes happening and how the kinetic energy informs the processes here, the soft landing ratio, but also chemistry on the surface. And you see these machines don't have changed since then. So you have some ion source with some collimating ion optics, mass filters, lenses to guide them around, and then some sample transfer, because when you make these sophisticated samples, you need to get them under control somehow. So later here, my concept, you will see, looks very much the same, only that the transfer here is directly aligned with the imaging methods I showed you before. So I hope that this underpinned a little bit where the problems come from and how we are aiming to solve them. I can, of course, now explain a lot about ion optics. I only briefly show my rundown about what is, in my opinion, crucial to get under control and possible to be under control. So you have your mass filter to pull out individual ions or regions of interest. You build sample holders in such a way that you can measure current. And this really allows you everything control further down. When you can measure current, you can measure beam energy, because you can apply bias voltages, and as your ions don't reach your surfaces anymore, you're higher in energy than the kinetic energy of the ions. Hence, you know the kinetic energy and can adjust. And at the same time, you can measure coverage because each ion carries a charge, and charge arriving here is measured as current. So you know exactly how much you place. So current measurement and mass measurement are really crucial here. And so now very briefly, a few examples. And for scanning probe microscopy, I'm really proud to say that this method opened SPM to all types of molecules. So chemical synthesis and biology is so prolific. And now we can look at all these molecules here, biological ones. This I did not want here. Okay, good. Biological ones like proteins, peptides, glycans, but also synthetic ones. And I'm especially proud of this measurement down here, which is a single molecule magnet. And when you place it in the tunneling microscope at 1 Kelvin, you can measure magnetic excitation here. Markus Ternes and his team did this. And you can see, so the outermost here is the spin flip, and the two inner ones is angular momentum projection change of the giant spin 10 complex in there. So this is the level of detail you can go down with the tunneling microscope. At the same time, you can do this with these huge methods, and I think this has a fantastic perspective. You can do chemical reactions, and you can look at them. Here is an imaging of a collision reaction that leads to different fragments, and we can track this over energy and really understand the chemistry that happens in these collisions. Again, by looking at the molecule, we have enough information to play out the entire story. And I want to especially highlight my colleague Kelvin Angara, who basically took over my old lab in Stuttgart at Max Planck and pushed these instruments there to the limit, imaging all these glycans and glycoconjugates here. There's all these different examples, and very well deserved. He has this beautiful science paper where he shows what diversity of information you can gain from this. I think the highlight is to directly look at a protein and identify glycosylation sites. And this really moves an imaging technique, which was before you saw it. So pentacene was the highlight. Everyone, I would argue, everyone knows how pentacene looks like, but knowing how these things looks like is really impactful. And there you see how these techniques fit together. So in the last 5 minutes, I want to say a few things, mostly reiterate what Josh said about native landing, and maybe give you my take on all this. So you can, of course, use native mass spectrometry with these very fine tips to get folded proteins into the gas phase. And I think it always has been an important question to say, what are these structures? So you heard Charlotte refer to us. Thank you. Very, very proud that this is relevant. So we now know how proteins look in the gas phase. You saw from Josh, they are folded because native mass spectrometry is really efficient. So when you start looking how many molecules are nowadays measured and what information people extract from native mass spectrometry, there is a lot of chemical information there. And similar, like for scanning probe microscopy, it would be wonderful to underpin this with structure directly linked. So here, these two viruses here from Albert Hecht's lab, or a protein complex. So these are first measurements from Carol's lab, I recall, or these interactions here of these proteins, and then directly see the structure. So not using theory to figure out where a lipid is bound, but seeing the bound lipid in the structure when you extract these peaks. I brought along the starting point for this because thinking about how long we are doing this. I think this was in 2016 or 17 when Joe Gold, who's in the audience today here, came to visit us in Stuttgart, and he did a first attempt with proper native mass spectrometry and some staining on the workbench. And we cherry-picked a few molecules, and we saw, yeah, this might just work. And because before we did this here with the holography colleagues, where we saw also molecules deposited on these graphene sheets in holography that had roughly the right shape, but our native mass spectrometry looked quite awful at the time. So Joe fixed this problem for us. So he showed us how really to do this. And before I come to electron microscopy, I want to feature here the colleagues of the holography in Stuttgart. So they built also their new instruments, and they are also using native mass spectrometry to look at really floppy molecules here, these antibodies, or what this is, transferrin here, I think, molecules that are really highly mobile in the gas phase. So you want to look at the single molecule. So for electron microscopy, you have to think about a lot of things again, because it's not a UHV technique. It's not about the vacuum and the purity. It is really about this part, getting the molecule in the gas phase in a good shape, landing it carefully, transferring it, and perhaps embedding it in a matrix that we need to learn in order to get good structures here. And by the way, we are not competing here with a method which cannot handle molecules like this at all. Cryo-EM works, and works very well. So we really have to add something to the table here. So our take on this similarly is a Orbitrap converted by a beam column, and we paid also special attention for this sample holder with a cryo shuttle, which can shield and control the temperature for this deposition. So the performance of this instrument you can see here. So if you have a well-behaved spectrum, you can indeed pull out a single peak. It can be more complicated for these more complex spectrums. But usually we would pick out here a region that cleans up the spectrum nicely. It is very important that you need intensity. You want to deposit a lot of proteins, so we change the interface. And the important thing is the energy, not necessarily the energy of the beam itself. This can be adjusted. It's the energy width of the beam that you need to control in order to get gentle conditions, because you see this width informs you of the maximum energy your particles will collide with the surface. So here again, Kelvin calculated this once, what difference it makes when you collide with 5 eV per charge or 50 eV per charge. I think it becomes clear and apparent that energy plays a role in retaining the fold on the surface. The surface itself plays a role. You can clearly see when you work on graphene, you get diffusion, so not on amorphous carbon. This might also depend on the temperature, and you see again here the structure 2 years ago. So what Josh showed you, very good structures, really nice native shape. Some resolution missing at the time. I thought this is maybe from the staining. Probably they are fine. Whereas we saw some disorder when we just deposited by room temperature and image in cryo-EM without ice. So what helped us in the end was this workflow here of depositing on the cold surface and being able to grow ice in vacuum, similar as Josh proposed, and then directly transferring into liquid nitrogen. So here are the samples without ice and with ice, and these are the results. So resolution you can achieve in the range of 2.5 angstrom, and this is enough to assign the models. So this is how these models look like. So the PDB structure, then when you morph it into the cryo-EM structure after soft landing, you see that these cavities are closing, and we understood this with MD simulation to be the consequence of dehydration. So yes, indeed, adding a matrix in some way and controlling this matrix is the crucial part in order to control your structure. So I always like to point out how many water molecules you still have when you have a dehydrated structure. So the MD here shows you 1,400 water molecules, but this is already dehydrated. And so to summarize all this, so yes, embedding is important. Secondary structure is quite resilient. Also, the domains are resilient, but they can reorient. So the bigger structures of the protein can shift around when you dehydrate them. But the method is quite reproducible. So here we made a series of measurements with this beta gal. So this looks all quite nice. And if you take on other molecules, from GROEL and C-reactive protein or ADH, then you notice in each case you get in the resolution of this final structure here below 5 angstrom, and this is where you can build the models. You see here GROEL and ADH change a little bit again as dehydration, whereas, for instance, C-reactive protein is extremely robust and stiff, and we almost see no changes. It is very positive, and we're very happy that we also see good results from membrane proteins. They even seem to be a bit more robust. So people say, oh yes, they are hydrophobic, they like vacuum. I think that's not even wrong, but there might be a bit more to this story. We have more parameters to control. We want to control now how many surfactants you carry around, and this makes the whole thing again more complicated. But also this seems to work, and with this I want to end here, pointing a few things out. Of course, we need now to think and build next generation instruments, and I think Charlotte's machine shows what is interesting and relevant and what could be done, and we'll see if all of this works at the same time. But of course, more intensity, more selectivity, mobility filters, controlling sample environment are the key points here. Moving into other fields, again, I'm always interested in this hyperthermal chemistry and using these ion beams for something else. And if I look in the world and almost count 10 machines sitting around or working currently on this, which makes me very happy and very proud. So here a lot of people helped me here, my group, especially Tim, with who I worked, and Calvin. Financial support, especially Thermo, supported us relentlessly in the past few years, as well as Carol and her lab here working with us. So with this, thank you very much. I hope we did overstay the welcome, not too much. Thanks for having me.
Thank you very much, Stephan, for that fantastic talk. To open up the questions, I had a few for you, if you wouldn't mind. First, you showed towards the end of your presentation a couple of high-resolution structures I hadn't seen yet for GROEL, for example.
Yes. Yeah.
There's been some open questions in the native mass spectrometry community about the gas phase structure of GROEL, and your model seems to suggest it has a fully formed cavity.
Yes.
I'm just wondering how much you've compared your structure with these other measurements suggest that GROEL in the gas phase collapses a lot more than you might expect it to.
Brandon, to be honest, I was quite surprised by this one. I expected the soft mode to be the how could we tell this distortion of the barrel, not along the axis. This is what we saw right now. Normally we simulate this, so and to understand why it is soft in this direction or what changes there. So I don't want to speculate a lot. It might also be so several things can happen, and always think about we are imaging a population that is absorbed on the surface. So for instance, if the surface adsorption causes a damage, it would come out in a specific region more than in another one. It could be this, for instance, or this could happen in the gas phase. So all of these things we have to understand. So, but I was surprised as well. Yes, but what you still see is, again, you can see the alpha helices. They are there. The cavity is there. So it is really about the top and bottom end of this barrel. And when I think about that, this is also the binding end. I think there's a cap that can close. So maybe this is the fragile part, right?
Mm-hmm. We have a question that's come in over the Q&A tab from Nicole Wagner. She says, related to my previous question for Josh Coon, instead of using a quadruple for charge state selection, would I-mobility separation of charge states be easier to implement or effective? Like a TIMS device in the front end, perhaps even with a device capped at cryo temperatures to preserve water molecules?
Yes, it would be nice, but I think not easier. As clearly pointed out, a TIMS device alone does not mass filter sharply. It just is a high pass or low pass, depending on the gas flow direction. And other mobility filters are not continuous beam, so they would lose a lot of energy intensity. But certainly, mobility filtering would be wonderful. I think we see this in native mass spectrometry. Multidimensional filtering gives you just more information, and it would be beneficial here as well, but not easier.
Absolutely. Liz, do you have a question?
Yeah, I do. As a non-mass spec person, I'm curious about a couple of things. So these distortions that you see, is there potential then to leverage that to understand the structure better? Do you know what I mean?
Yes, absolutely. I think they tell us something. So as I just said about the GROY-L, they in my opinion, they indicate, I call this a soft mode. So each molecule can vibrate, and where the vibration is the easiest, the softest, interestingly, with lowest frequency, this is where the molecule can deform. So I think that's what we are seeing. So for instance, for antibodies in this holography experiments, we always saw them clumped together in a structure, in a collapse structure. So now colleagues told me this is maybe not a gas phase collapse. They saw in it actually a functional collapsing mode of the molecule. So yes, there is information in it. We have to learn how to understand it. We already know that we can manipulate this. So in the holography, we always use the energy to partially open these structures again. Could be that this is a little bit too violent, but yes, there is mechanical. I see the mechanical properties of the molecule in there, and again, we have to learn how to control it to understand this. Yes. Yeah.
I guess to follow on from that, because it's kind of, you've already hinted at it already with understanding different conformations that are present, right? And continuing to parse out all these different bits of information that are there. Where are things at with maybe understanding something about, can you comment on dynamics? Things, different conformational changes that are happening in solution that you're capturing in these methods? I know maybe that's too big of a reach, but I'm actually amazed and thrilled at all the things that have been happening as a non-mass spec person, but coming in and seeing what you're doing. There's so much more we're going to learn, that's clear.
So for electron microscopy, we have not done this. With tunneling microscopy, I did not show this today. We did this already many times. So yeah, we use this quite deliberately, that we use collision with the surface or in the gas phase to manipulate the conformation of a molecule, and then we can measure the outcome. So we can activate molecules and have them stretched out, or we are gentle and they are in a coiled up conformation, and this becomes very quantitative. This can be done with the surface. Then you excite a soft mode. You can do it in the gas phase. Then you have more gradual heating. So this is the beauty of these vacuum-based methods. The drawback is you're not immediately native anymore, but what happens, you can really control it. So you have more information, and now the interplay will be how to be native enough to learn something about biology while still be in control of all the things that happen.
So we have another question that's come in over the Q&A tab. This is from Martha Ortega. Were the MD simulations done with completely dry complexes, or were some bound water molecules included, as was observed experimentally according to your presentation? If so, what was the criteria for choosing where to place the water molecules?
Yeah, I'm sorry, I rushed through this a little bit, so I can add a little bit more detail. So the simulation worked such: we took the PDB model, we solvated it completely, we relaxed it. So this is the first bit. Brandon, can we see the screen still? Yes.
We don't have your slides up at the moment.
Okay.
But maybe we could.
Okay. Good. Okay. So then in the first bit we relax it. Then we have a proper solution structure. Then we place it in a droplet, and then there's a trajectory stitching, always removing more and more water. And this is how we move to a desolvated structure. So it is really a simulation of a desolvation. And this one reaches then a very similar structure as we found in the experiment. And then we resolvated it again, and it returns to a more or less native structure. So this seems to be the real deal of hydration and dehydration that we observe here.
I guess I had one last question for you, I guess, and I guess unless there's other questions that come in over the Q&A or other people have. So I wanted to ask you something similar to what I asked Josh about the future of this really exciting and dynamic and fast-moving field, right? There seems like there's all sorts of things that need to be done and lots of different directions that we could go. And I'm just wondering, what do you think are the most important, immediate objectives to try and focus on right now, especially when it comes to trying to convince the broader structural biology community that there's something that's worth paying attention to here?
I can honestly tell you, I always approach this from instrumentation perspective, and this is where I see this. I wrote my list before, so it's intensity, selectivity, and controlling sample environment. I think there's a lot of things to be worked on, and then the results will become even better. I'm convinced about this. What the structural biology colleagues want to see, and they basically told me in Memphis, where I just came from, they told me they want to see sort of new value brought to the table. So show us a structure that we can otherwise not get, pull out an interaction that we can otherwise not pull out. So they are quite pragmatic, annoyingly pragmatic, but not wrong in what they are saying. And the way to achieve this is, I think, these technical improvements.
Absolutely. Well, we have one more question that's popped in, and this might be the last one we're able to get to today. This one's from Federico Carrion. Federico says, I'm very impressed with the glycopeptide images. Do you think you can be confident about the backbone structure due to glycan's restraints, or is this more related to the artifactual deposition process?
Okay, so I'm not sure I 100% understood this.
I can repeat if that's good.
I have it. I opened it myself, Brandon.
Okay.
Are we confident about backbones? Okay. Yes. So yes, the backbone of peptide or the glycan. Okay. Good. Let's put it like this. So yes, we are confident about these. You can distinguish protein and glycan just by size. The sugars are bigger. They are more three-dimensional. The proteins tend to lie flat on the surface. The sugars are bigger, so you know what is what. And then you have chemical restraints, just steric restraints, which don't, once you see the blobs, we call this blobology always, once you see the blobs plus the steric restraints, don't give you a lot of freedom anymore how to place your molecules there. It becomes then a puzzle of DFT and molecular dynamic to get the actual structure. Going further from this, we just have this accepted in Nature Communications. We recently showed that you can image this with non-contact AFM. Then you go up a whole step in resolution and you actually see the location of OH groups and hydrogen bonds and can quite confidently assign structures. I hope this answered the question.
Okay, great. It's just about 11 a.m. here on the East Coast, so we should maybe bring this to a close. Thank you so much, Stephan. And thanks also to all the rest of our
Great to
speakers,
see all the
Josh
colleagues.
and Charlotte, for a great webinar. I feel like, since Stephan mentioned this small meeting that Josh and Stephan and I helped organize, it's worth mentioning to this audience, especially if you've stuck here to the end, or if you're watching this on the View on Demand. There is a small meeting that covers this topic called the MS+M meeting series. The next one of these will be in Utrecht in 2025. If you're interested in this subject area, I highly encourage you to attend. And maybe also you'll see this topic pop up at a future Protein Society meeting. So I would encourage you to do that as well. Liz, do you have anything to add at the end here?
Well, we'll just wrap up. This was a really exciting webinar. Thank you all so much for sharing all this with us. And everybody, if you are sort of learning more about it, you'll see this also posted on YouTube. So I encourage you to go there, because I think we need to grow what's happening in this field. So thank you.
Agreed. Agreed.
I
All
think
right.
this is a
Thank you,
wonderful
everybody.
topic. Thanks.
Thank
Good.
you.
Bye-bye.